So today we're going to focus on transcription and then how that RNA is processed, in particular in eukaryotic organisms, before it continues on to translation. So we'll have a separate video on translation, and so our focus today is just the transcription, the DNA to the messenger RNA. So a few reminders about RNA. So RNA function, just like DNA, is determined by the sequence of those RNA bases. And so its function is a little bit different than DNA's. I know we said that it could carry genetic information. Okay? Usually it's helping with the process of protein synthesis. So its structure is going to be a little bit different than DNA. It's going to have ribose sugar instead of deoxyribose sugar like DNA has. Its nitrogen bases are going to be a little bit different. Remember, we have uracil instead of thymine, so that U goes with A and C goes with G. Remember, if I have DNA strand that has an A or a T, when I'm making my RNA, it's a U, and the T binds with an A. So U just takes the place of T. It never binds with T. But our C and our G are the same. Remember, our pyrimidines are cut from purines. So my pyrimidines in this case is uracil and cytosine in RNA. It is single-stranded, not double-stranded. You can see that over here. Remember, DNA is in our double helix, okay, where RNA is a single-stranded molecule. And there are multiple different kinds of RNA. We have messenger RNAs, and that's my mRNA, tRNA, which is my transfer RNA, rRNA. We have siRNA. There's a variety of RNAs. You don't necessarily need to memorize all the different kinds. We're going to talk specifically about messenger RNA and transfer RNA the most because they're the ones that are the most heavily involved in transcription and translation. Remember, our basis of transcription is taking that DNA and turning it in particular into mRNA or messenger RNA. So transcription is going to be the process again of taking that DNA and turning it into messenger RNA. So I'm going to add nucleotides right here and create a messenger RNA strand. I'm not actually turning DNA into messenger RNA. So when I unwind the DNA, this area right here is what's called our transcription bubble. And so the transcription bubble is just this small little part of DNA that's unwound at a time so that you can make messenger RNA. So it's where the actual transcription is happening. It's not a literal bubble. It's a bubble because that's what the DNA looks like as it unwinds, because we're not unwinding large amounts of DNA at a time. Okay? The DNA is going to unwind a little bit. Will make the messenger RNA. It will close back up and new parts will open up. So the DNA strand that is actually transcribed is considered our template, our non-coding strand, our minus strand, our antisense strand. There's a lot of terms for it. Template is going to be the most common. So for instance, in this case, this one right here is my template strand or my non-coding strand. And this untranscribed DNA, this one over here, that one is going to be my coding strand. Because if you think about it, when this messenger RNA is created, the messenger RNA is created by using complementary base pairing, right? So if this is an A and a T and a C and a G and an A, okay? So if those are my bases there, then my messenger RNA is going to be a U, an A, a G, a C, and a U. Well, my DNA on this other side is going to be a T, an A, a G, a C, and a T. So this coding DNA should actually match my messenger RNA. So that's why that one is called my coding DNA, but the other side that is actually the side that's getting transcribed is considered my template or non-coding DNA. Okay, so my coding DNA is going to end up having the same sequence as my messenger RNA, and that's my goal, right? That's how I made a copy. Since I can't make a direct copy and I have to use complementary base pairing, this allows for the copy. And the enzyme that's going to accomplish this is going to be RNA polymerase. When we were talking about DNA replication, remember we said that DNA polymerase is the one that added more nucleotides. So in this case, I'm going to have RNA polymerase adding these messenger RNA nucleotides onto the strand to make it longer. And again, all of this is going to be happening in that transcription bubble. So we have multiple kinds of RNA polymerases. We have 3 kinds in particular, RNA polymerase 1, 2, and 3. Every single one of them is going to be adding nucleotides, but they're just going to be making different kinds or transcribing different kinds of RNA genes. So for instance, RNA polymerase 1 only transcribes rRNA genes. So it only transcribes information for rRNA, and rRNA is ribosomal RNA. So it is used to actually make ribosomes, which are important for protein synthesis. RNA polymerase 2 will transcribe genes into messenger RNA. So this is the one that we are thinking of usually when we're talking about RNA polymerase. So this will transcribe genes, the genes that are going to be turned into messenger RNA. It, just like with our DNA polymerase, it builds in the 5 to 3 direction. Okay, so when my DNA is unwound, and I've created that little transcription bubble here, if this is my 3' and my 5' end of DNA, so that would make this my 5' and this one my 3'. Okay, RNA is built the same way. It will build only in that 5 to 3' direction because it's reading it the other way. So RNA polymerase 3 is going to only transcribe tRNA genes. Remember, tRNA is our transfer RNA. That's important also in translation, helping move those amino acids. So again, the one we're going to most commonly be referring to would be RNA polymerase 2, because we'll be making messenger RNA. So in eukaryotic cells in particular, you're going to have various regions along the gene, and this is how the cell is going to help know where one gene starts and one gene stops. So for instance, we're going to have this promoter region, and it's going to promote, or it's going to start the gene. So the promoter region is going to occur right before the gene, and it's going to be a binding site, and it's going to consist of what we call a ta-ta box. And so the ta-ta box is the binding site for these transcription factors and the RNA polymerase. Basically, it allows the RNA polymerase and the transcription factors to attach before the gene gets going, before you actually get into the meat or the information of the gene. So this ta-ta box here, located within the promoter region, is the signal that it needs that it can attach there and it can begin transcription. The enhancer region is located upstream of the gene. Okay, so by upstream, I mean it would be located further down the end. So it is a binding site that is going to be located way upstream of the gene, so kind of away from the gene on the DNA. And that's going to kick your transcription into high gear. So when factors bind to that enhancer site, that's going to speed up that transcription process. So you can do lots and lots and lots very quickly. So once this messenger RNA is made, in particular in eukaryotic cells, it's going to go through some modifications and some changes. Okay, so all of those are going to be controlled by enzymes, and one of those changes is going to be that we're going to need to remove introns, or what are called junk DNA. So prokaryotic organisms won't have these introns, but eukaryotics will need to do this. So eukaryotic genes are not continuous. So we have parts of the gene which are called the exon, which is what's actually expressed and coded for, and then we have parts of the genes which are called the introns, which are the junk DNA, or the spaces in between the different coding sequences. So these introns will be cut out and removed, and this is beneficial. It seems like a waste of time, but it is beneficial in the sense that it allows us to rearrange these exons into a variety, into a different variety of proteins. So basically, I can get a different variety of proteins from just using one gene by doing the splicing and the rearranging of my exons. Okay, but this is important vocabulary that you get situated. Exons are the ones that are expressed that we show. Introns are the ones that are considered junk that we get rid of. So this is called post-transcriptional processing. the splicing of the removal of the introns and the retention of the exons. So the splicing is the cutting and the removal there. The retention is the holding on to the exons. So my post-transcriptional processing, so after transcription, the first thing that I make is what's called my pre-RNA. That's this one right here. And this is happening on eukaryotic cells only. So the eukaryotic cell has made this messenger RNA based off of the DNA. The top one here is my DNA. And that pre-mRNA contains those introns. And so we will go through and it will splice out these introns. So it will remove those introns. And then it will edit those exons back together. And I don't know why the picture is cut off, but what you would see basically is all the green ones would be linked together down here, and that would be my spliced messenger RNA. That's my actual mature messenger RNA. And that is what is actually going to go out into translation and actually turn into the protein. And so the way I cut those introns out and put those exons back together can give me different versions of that mRNA. And if I get different versions of that mRNA, then that allows me to get different versions of that protein from the same gene. So for instance, if we look at our picture here, we've got our DNA at the top. Okay, and then there's our RNA transcript right here, the second one. And then based on the way the RNA was spliced together, I can get a couple of different possibilities for my final messenger RNA, which can lead to different possibilities for the proteins. So this could happen when an intron is not really considered an intron. So maybe it isn't considered junk DNA for that particular version of the gene. Okay? I can also treat different segments as my exons depending upon the various the different versions of the gene. So for instance, if I look at the DNA, all the blue ones are exons, and so are the green and the red one. But I have in my two different versions of my final messenger RNA, I have one where the red has been spliced out and one where the green has been spliced out, giving me alternate messenger RNA versions, which will give me alternate proteins. In addition to the removal of those introns and the splicing back together of these exons, there is some more messenger RNA processing that needs to happen in eukaryotic cells. So again, these are modifications that are done to that messenger RNA before it goes out to finish doing translation. Okay, these are done to help protect the messenger RNA as it goes out to the cytoplasm from the nucleus. So we add what's called a poly-A tail. Poly many, right? So just a tail with lots of adenines. So we add a poly-A tail and we add what's called a GTP or a 5' cap. So my GTP and my 5' cap and my poly-A tail, both of them are used to help protect the messenger RNA from being degraded or broken down as it moves out through the cytoplasm. So one of their jobs is helping to protect the ends of that messenger RNA so that if anything starts getting broken down or dissolved, basically, it's not the important part of the messenger RNA. So the 5' GTP cap will help one end, and the poly-A tail will get added to the 3' end. The longer my tail, the longer that messenger RNA will last. The longer the messenger RNA lasts, the more of the protein that particular messenger RNA will make. Okay, so the poly-A tail is important in addition in general degradation of the messenger RNA. It also helps dictate then how long that messenger RNA is going to continue to make proteins for. The 5' cap, in addition to helping protect the 5' end, is also going to help the messenger RNA attach to the ribosome. So that is an important function in it as well, because if it doesn't attach to the ribosome, then translation won't occur. So this is all part of transcription, taking that DNA, making that messenger RNA copy, and then modifying that messenger RNA copy, in particular in eukaryotes, so that it's ready to actually do translation. So we'll work with this stuff in class, and then you'll have a second video that focuses solely on translation.